So what exactly happens is, these are this thing, okay. Now there are different types of B lymphocytes and T lymphocytes and so on. So we'll also discuss a little about this. Okay, somebody asked me about the difference between monocyte and macrophage, okay. So the main difference between monocyte and macrophage is this: both of them are phagocytic cell, okay. Both of them are which kind of cell? Phagocytic cell. Monocyte is a phagocyte, and then macrophage is also a phagocyte, okay. But the difference is that the macrophages are residual. For example, Kapher cells, okay. For example, what? Kapher cells. Kapher cells are kind of macrophage only. Then there is alveolar cells. Alveolar cells, okay. And then there are some more. So these Kapher cells are nothing but the macrophages of the liver, okay. Macrophages of the liver is known as Kapher cells, whereas the macrophages of lungs are known as what? Alveolar cells. Alveolar, it is not alveoli. We are not talking about this. We are talking about alveolar cells. That means inside the alveoli, whatever macrophages are there, consider this: you are inhaling a droplet which contains a lot of M. tuberculi, okay, or like today's case, like the COVID virus, okay. So what will happen whenever this droplet enters? There should be somebody to look after the immunity of the lungs, okay. At least there should be some non-specific activity that will be going on. So who will look after this non-specific immune response or protection at the localized level, local level? It is what? The macrophages, okay. Now these macrophages are staying where? Inside the lungs. So what will happen if some kind of blood infection takes place, okay? A systemic blood infection takes place. So who will look after? It will be taken care by this, what we call as monocytes. So monocytes are again monocytes are again macrophages only, but they are circulating in the blood, okay. And then these eosinophil, neutrophil, basophil, mostly these are phagocytic cell. And in eosinophil, as you all know, it helps in allergic response, okay, mainly against parasites. Now it can also be for some other kind of this thing like tropical eosinophilia. So in that case also there will be a lot of eosinophil, okay. But mainly eosinophil, the number of eosinophil increases due to an immune response against parasites, okay. So TCTC is again important this thing to discuss. We have not yet entered into the typical immunology. We are like in the periphery, we are moving around. So whenever there is an infection, you talk about total count and differential count, isn't it? Why this? Because you understand that wherever there is an infection, so this kind of cells will increase in number, and eventually it will give you an idea whether the patient is at least trying to overcome the infection. Many a times what will happen, if the patient is already in the shock, then what will happen? You will see that there is no response. The infection is there, you know about it, there is significant tachycardia and so on, but what you are seeing is there is no change in the total count, there is no increase in specific granulocytes and agranulocyte numbers also. But eventually you also see one more thing, that there is a depletion of platelets. So in that case also you can understand that the patient is not, the immune response is not up to the mark or suboptimal. So this total count, differential count is a very important parameter which will help us to identify what is the condition. Then some other aspect of this is that okay, now B lymphocytes. Now B lymphocytes are very important. Why? B lymphocytes are the cells which will look after the humoral immune response. Okay, B lymphocytes. So B lymphocytes are responsible for what? Immune response are of multiple different types. Okay, so I'll come to that point. So B lymphocytes will look after humoral immune response. I'll write it like this. IR means immune response. And one more type is called cell-mediated immune response. Okay. So how these are different, that also we should understand. Cell-mediated immune response. Okay. So let me tell you this. Whenever there are some foreign invasion in the human body, which are like bacteria. Okay, some kind of bacteria has entered through the this what do you call aerosol or the droplet, and it enters into the lungs and then eventually entered into the blood. So what will happen? Humoral immune response will be initiated. Now this initiation is taken care by means of B lymphocytes. Those lymphocytes which have been originated in the bone marrow as well as matured in the bone marrow itself. Okay, so let us see how exactly this B lymphocyte will look like. Now in B lymphocyte you will see a typical structure. Okay, so we'll have a look. Okay, so in B lymphocyte you will see something like this. Okay, on the surface. Okay. Okay, I am talking about like if you do a electron microscopy, then you will see that on the surface of the B lymphocytes there are like, what shall I say, this surface receptors. Okay, which will look like antennas. Can you see like antennas? Okay, like this antennas you will see. You will also see one more thing which is called, I cannot able to draw, I think here. On the surface there are multiple different types of antigens. Multiple different types of antigens on the surface of the B lymphocyte. It's very typical for B lymphocytes. Okay. Now there is also a nucleus, a prominent nucleus, not like granulocytes. In granulocytes there will be different types of segmentation. For example, in eosinophil you will see tri-lobed nucleus, whereas in basophil there will be a single U-shaped nucleus. Nothing like that. It's a firm round kind of nucleus. Okay, ekdam round. On the surface there will be receptors, or shall I say what? Shall I say this what do you call is surface antigens. Now these surface antigens are nothing but what? These receptors are also known as antibody molecules. Okay, so what is the function, major function of B lymphocytes? The major function of B lymphocyte is to what? Generate antibody. Now these antibodies are residing on the surface of the B lymphocyte. Now one more antigen is also present on the surface of this what you call B lymphocyte, which is called MHC1 molecule. MH, very important, MHC1, as you all know. This is responsible, but what you do a MHC screening, no, before going for transplant and all such things. We will come to those discussions also, but in the beginning you just understand that on the surface of the what you call this B lymphocyte there are several antigens. Okay, so one antigen is called the antibody itself, so it itself behaves as an antigen. Okay, and there is MHC1 molecule in one side, in another side there is an MHC2 molecule as well. So this is very, very typical for what? B lymphocyte. MHC2. So MHC1 antigens are there on the surface of the B lymphocyte. At the same time there are MHC2 antigens, and also there are antibody molecules. Okay. 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 Is there any chat? Just hold on. We'll see. There are some relevant discussion there. So these are the things which okay. Bursa fabricius is basically, that is right only, but what exactly happens I will tell you. Now Bursa fabricius